My job now is to introduce our keynote speaker, Dr. Bruce Cheson. On your table, you've got a list of his amazing accomplishments. So I want to just tell you three things. Remember a little while ago I talked about IWWM8, that workshop that Dr. Treon's developing in London? There's going to be 250 researchers there. Well, back in the year 2000, September 2000, Dr. Bruce Cheson was the instigator behind the very first one of these, where he had 19 researchers, and they came together to discuss coming to consensus on a basic understanding of WM, how it's defined, how it's diagnosed, and on what the prognostic factors would be. They didn't agree, but they did agree they should meet again. And they've been meeting now. This will be the 8th one. And without Dr. Cheson, it wouldn't have started. So let's give him a warm round of applause as thanks before he even gets up here. Now, Dr. Cheson was at the keynote speaker at the LRF Educational Forum that some of us attended last fall in New York. And he was incredibly good, incredibly good. And Guy Sherwood, who's our Vice President of Research, said, wow, I'd really be impressed if he came to speak at our Ed Forum. So guess what, Guy? You and everybody else are ready to be impressed. Get ready to be impressed and learn and enjoy Dr. Bruce Cheson.
Forward is right and backwards is left?
Yep.
I just had to bring my vitamin W with me. Hope it doesn't fall. I just have to hold it and keep drinking it. Thank you for having me. It's a pleasure to be here. I do want to single out one person here because I'm kind of a nothing compared to the one person in the audience who's probably, not even probably, certainly one of the greatest hematologists of all time, and that's Bob Kyle. He's over there. Here. There are few that have made the kind of contributions to our knowledge of myeloma, Waldenstrom's, and other diseases like Bob, and I'm honored to even be able to sit next to him. So tonight I want to take you through a little historical perspective on progress in indolent lymphomas, of which WM is one, and ask the question, is chemotherapy dead? Now, over the years, we've had a number of treatment approaches for cancers, lymphomas, leukemias, etc. We had surgery where we sliced them. We had radiation where we fried them. We had chemotherapy where we poisoned them. And now we have targeted therapies. And where do we get chemotherapy? Well, chemotherapy actually dates back to WW1 and 2, when we had mustard gas. And what they found was that when the Germans dumped mustard gas on the rest of the world, it caused skin ulcerations, blindness, lung damage, nausea, vomiting, and caused cancers. It was chemical warfare. But what they also found was that accidental exposure led to low white blood cell counts, and the thought occurred that it might have similar effect on cancer cells. So in the 1940s, these chaps named Goodman and Gilman, who wrote a famous textbook that we all used in medical school, gave intravenous mustard gas to patients and in fact found that there were responses. Lymph nodes got smaller, spleens got smaller. The responses were short, but it led to the subsequent development of other less noxious substances. And this is just a short list of chemotherapy regimens we use for various lymphomas. And the orange are various alkylating agents. So they're still around all over the place for Hodgkin's, for non-Hodgkin's, leukemias, and others. Then we took a bunch of drugs and we threw them together haphazardly and created chemotherapy concoctions. And this is a very famous slide that we in Lymphoma Land show. In patients with a disease called diffuse large B-cell lymphoma, which is the most common of the non-Hodgkin's lymphomas, we took the original regimen called CHOP, and we added things, we mixed things, we made all these algorithms of various chemotherapy drugs, CHOPs and BACOPs and MAKOPs and all this. And in the end of it, they were all the same. So we were making them more toxic and more expensive, but more wasn't better. And so CHOP remained a household word in lymphomas for many years. And then some believed that dose intensity was important, so we doubled the dose of CHOP. And we had the CHOP regimen, and we also found that you could give it to kids. There's a pediatric featuring the new kids menu. We added other drugs to CHOP. And we even tried to give it as an oral preparation. But in the end, it was still CHOP and it was still toxic. So we want to get rid of chemotherapy, and for the last decade, that has been my goal, is to be getting rid of chemotherapy in patients with lymphoma because it has non-specific effects. It not only affects the lymphoma, but you lose your hair, it affects your stomach, your skin, your fingernails. So it has acute and long-term toxicities. Many patients can't tolerate it, and I defy you to raise any hands in the audience if anybody really wants it. We have new targeted agents, and what I'm going to show you is that there are biological combinations that have activity comparable to chemotherapy and chemoimmunotherapy. Now, the concept of immunotherapy is relatively new. It started back in 1854 with Paul Ehrlich, the father of modern immunology. And Professor Ehrlich said, you see, we must take aim by chemical variation. The marvelous effect of an antibody in the serum is due to the fact that in no case it has affinity for the body substances, but flies straight to the parasites. He created the term the magic bullet, the monoclonal antibody that killed bacteria, killed tumor cells, and didn't affect the normal body cells. And this is a monoclonal antibody. This is what it is. You've got the cell, and you've got those proteins on the surface. You heard about the CDs, that's them. And the antibodies fit like a lock and key. And they have been a marvelous advance in the treatment of leukemias and lymphomas. In fact, if you look at, again, large cell lymphoma, I showed you those curves that were all superimposable. Now you add this monoclonal antibody, rituximab. It's the first time we've been able to improve the survival of patients with large cell lymphoma. You see how the curve goes up for the first time. So there are important questions. Where are we going, and how are we going to get there? Well, we're going to chemo-free therapies and personalized treatments. And we're going to get there by a better understanding of the biology of the tumors, by recognizing patient and tumor diversity, and through good clinical trials. Let me give you an example. Perhaps the first good study that showed that we could get rid of chemotherapy was done by a Swiss group. They took rituximab and they gave it to patients with indolent lymphomas, and they randomized patients to prolonged administration versus not, and they showed that out at 8 years, there were still 30–40% of patients who hadn't progressed with just a few infusions of this monoclonal antibody. What we did starting a decade ago, we took 2 monoclonal antibodies and we put them together. And what we found was that in low-risk patients, 92% responded, 3/4 of them got a complete remission. Intermediate-risk patients, 80% and 50%, even high-risk patients, almost 60% responded, 1/4 got a complete remission, and their survival, you can see the survival curve, 90+% of patients are still cruising, 3, 4, 5, 6. I have patients out now 8 years who are still in remission, no chemotherapy. Well, we've taken monoclonal antibodies, we've tried to make them better. This is a drug called brentuximab vedotin, and it is used for Hodgkin's lymphoma and a form of T-cell non-Hodgkin's lymphoma, and it's really cool. You can see the monoclonal antibody, and it's bind to a poison. If you give this poison intravenously into the patient, that would be the end of the patient. But it's bound to the antibody. The antibody binds to the tumor cell. It's taken into the tumor cell, the poison is released and kills the tumor cell. Sounds good, better living through chemistry, but does it work? Sure enough, if you look at patients with this T-cell lymphoma called ALCL, or anaplastic large cell lymphoma, you can see that the response rate on the top line is over 80%. These are patients who have failed other chemotherapy regimens. If you look at patients with Hodgkin's lymphoma, who have not only failed chemotherapy, but who have failed a bone marrow transplant, the response rate's 75%. And a third of these are complete remissions. No chemotherapy. And most people here are probably familiar with a slide like this. These are the new exciting drugs. All B-cell malignancies, including WM, have a B-cell receptor on them. And when this is tickled, a number of pathways in the cell get activated and they make the cells immortal. They make the cells live. They make the cells thrive. They make them resistant to chemotherapy. But now we have drugs that target various of these pathways. The one you'll hear most about here is ibrutinib. And I won't mention much about it because you'll hear a lot about it, but I'll show you once that if you give the right dose, which is on the right, almost 60% of patients with indolent lymphomas will respond to this drug. God bless you. Gesundheit. And you're all now familiar with the Mighty 88 that Steve Treon published a wonderful paper in the New England Journal a couple of years ago. It sits right next to BTK, Bruton tyrosine kinase, which is the target of this drug. So it makes sense. Not only does it make sense, but it works. And I'm not going to show you WM data because you'll see plenty of them at this meeting. But there's another drug called idelalisib. And this targets a different pathway, the PI3 kinase pathway. And this has about a 60% response rate in relapsed and refractory indolent lymphomas. And this is what's called a waterfall plot. It just shows you the decrease or increase in the amount of tumor. The kind of cobalt blue lines are Waldenstrom's patients. And it just shows you that in the vast majority of patients, the amount of tumor goes down with administration. And these are oral drugs. These are pills. These are not shots. And these are not chemotherapy. The antibodies take care of the outside of the cell. Those drugs target the intracellular pathways. But the cells live in a microenvironment, a milieu. I love that word, milieu. A great word. Those French, they have the wonderful words, milieu. One of them is lenalidomide. Remember thalidomide. Lenalidomide is the next generation, and it has activity in a variety of histologies of lymphomas, ranging from 20–30% by itself. But it's a pill, and it works better with other things. Here's a study that we did a number of years ago. We combined, you can look on the right, I wish I had a pointer, lenalidomide and rituximab. 73% of patients responded to this combination who had failed prior therapies. And the median time, you show me where it is? Which one? I'm pointing at it. What's it doing? Oh, there it is. Hot dog. Look at that. The time to progression was 2 years. 2 years. And this is in patients who have failed real chemotherapy. And this is not chemotherapy. So what we did, we took this combination, which we had invented, and we put it in frontline patients with indolent lymphomas. And what do we get? Overall, this doesn't really work. 93% of patients responded. 72% had complete remissions of their disease without chemotherapy. And there is a very important trial called the Relevance study, which combines rituximab and lenalidomide or Revlimid, what we call R², and is comparing it with rituximab and chemotherapy. And this could very well change how we approach patients with indolent lymphomas. The study is almost complete. Huge international trial. Also better living through science. We're learning more about the microenvironment and that there are cells there. And there are things on these cells. They're what are called effector cells. These effector cells, here's the tumor cell, they're supposed to kill the tumor cell. But there are these proteins there, PD-1 and PD-L1, if this thing really works, you see it right there, which block these killer cells, put them to sleep. And now we have drugs which target PD-1, PD-L1, And the killer cells wake up and kill the tumor cells. And these are in clinical trials also. But we have a misconception about diseases. We think of diseases like a spider web, where you have a little spider and you cut it there and you're going to destroy everything, or maybe you cut it again, and but it's a little more complicated. We now know that there are a number of targets. We talked about BTK, we talked about PI3K, there are all these other ones. And yes, we can target one, and yes, we can target another, but it's a little more complicated than that. And so we have these drugs. How do we get the drugs from us out to you? Well, clinical trials. Clinical trials are critically important. And it's not a new concept. Clinical trials have been around for quite a while. In fact, they date back to the book of Daniel, where the health of the Hebrews fed a kosher diet was compared to the Babylonians fed a state diet. Any Hebrews in the audience? Any Babylonians in the audience? Point proven. Number 2, on the HMS Salisbury, they compared cider versus vinegar versus lemons. And they found that if you had lemons, you didn't get scurvy. No scurvy for me, right? You'll hear more about this, but I just want to throw a little bit at you. There are 4 phases of clinical trials. There's phase 1, which is a relatively new drug or combination, looking at the safety and toxicity in generally a small number of patients. Phase 2 looks at the activity in a larger number of patients, but still not a big one. Phase 3 is usually a randomized study comparing a standard to an investigational program. And phase 4 is what the companies do to market the drug and have increased indications. But we have a problem. There are more than 7,000 clinical trials open in the US, and only about 3% of patients go on studies. Studies compete. You saw there were 98 studies, but how many WM patients are there? They're competing for the same patient population. And 2/3 of clinical trials never get completed, either because it was too toxic, didn't seem to work, or you couldn't accrue patients to it. So problems in developing new drugs are the slow rate of accrual to clinical trials, lack of preclinical models. Giving a drug to a mouse is not quite the same as giving it to y'all. We don't have surrogate endpoints, meaning that we tend to look at survival, and fortunately in some diseases survival can be decades, but we can't wait decades. We need something to tell us sooner what works and what doesn't. There are false negatives. Sometimes we miss an active drug because we've just done it wrong. And there are false positives. We get really excited about something, we invest a lot of energy in it, and it really didn't work anyhow. Who does these trials? There are investigator-initiated studies. The National Cancer Institute sponsors studies. They also fund cooperative groups and cancer centers, and pharmaceutical companies sponsor them. And the cost of developing a drug is exorbitant. In the 70s, it was about $140 million. In the early 2000s, it was well over $1 billion to develop a new drug. But help is on the way. The FDA is finally smartened up and has a number of new mechanisms to speed the pathway. There's accelerated approval, priority review, fast-track designation, and what ibrutinib got, and idelalisib was breakthrough designation, which speeds their approval and appearance on the market. So why should we do clinical trials? Well, from the doctor's perspective, it attracts patients to the practice. We can provide cutting-edge therapy, potentially effective treatment options leading to improved patient outcome. All of those are very important. From the patient's perspective, you like to please us. You want to make your doctor happy because we've got you really intimidated. It gives you earlier access to novel agents. You can get free drugs. Ibrutinib costs about $130,000 a year. You get on a clinical trial and it is free. You get monitored very carefully, very closely. There are many layers of approval, so it's safe as it can be, It helps future patients, it helps make progress, and it gives hope to everybody. So when should we consider clinical trials? Until there are no more toxicities, until everybody is cured, we need to do clinical trials. Let's look at the pathway of drug development from my perspective. We have a new agent. Do a phase 1 trial. It seems to be safe, so you do a phase 2 trial to see if it works. It may have limited activity, in which case it gets flushed. Or it might seem to be active, and then you kind of just throw it together with something else that's also active, and you develop an empiric combination, but you don't know what the heck you're doing. So you've got to include the things you heard before, the pharmacogenomics, the molecular biology, so you know what you're doing. And to improve therapy and individualize therapy, you have to recognize tumor diversity, and you have to recognize tumor biology. Because the rule is that not all patients or diseases are created equal. Your Waldenstrom's is different from your Waldenstrom's, is different from your Waldenstrom's. Yours is kind of the same. Okay? And therefore, not all therapies should be created equal. Here's the point in hand. Antony van Leeuwenhoek, the chap who invented the microscope, we owe him a lot. It was around 1668, even before Bob Kyle. And what we now know is that you can look at a lymphoma under the microscope and it looks the same from patient A to patient B, but if you look at the molecular pattern, this is a gene profile. You can see there's red up here, green there, and red there. This is the pattern of gene expression, overexpression, underexpression. It looks the same, but it's remarkably different, and they behave differently. In large cell lymphoma, again, there are 2 different subtypes, an ABC and a GCB. I don't want to talk about them in detail, but even though it looks the same under the microscope and you treat them the same way, their outcome is very different. The GCBs do much better than the blue curve, which is the ABC subtype. So the better the treatment, the less relevant are prognostic factors. And the more relevant are predictive factors, meaning those targets on the cells that we can direct drugs to. Let me give you an example. This is chronic lymphocytic leukemia, another blood cancer. And if you do chromosome analyses on these patients, there's a bunch up here, and they all seem to do kind of the same. And then there are these that look like the ski slope at Snowbird. They just do terrible. Their survival, the average survival is like a year, 9 months or so, with all the standard therapies. And then along comes ibrutinib. And these are those 17p deletions now, with 70% of patients, a couple of years, still alive. So the 17p is no longer as important. Now in Waldenstrom's, there were these prognostic factors that have been published, and I can't see it for the balloons, but age and hemoglobin and the other things that you can see on the slide. And it divides patients into 3 basic groups based on the factors. There are the good ones and the not as good ones and the bad ones. But I submit to you that with these novel therapies, this will all be irrelevant because they will make everybody do better regardless. And they are not chemotherapy. Our issues for the future. How do we pick the patients most likely to respond to a particular drug? They're really expensive. You don't want to waste $130,000 on a patient who's not going to respond to it. So we have to figure out ways before we treat them based on understanding these pathways that I showed you. You can study BTK, you can study PI3K, and see if it's active and see if that correlates with outcome. So you have to tell which ones are likely to respond and which ones are unlikely to respond. Patients develop mutations in these pathways, which makes them resistant to these drugs. How do you treat patients who become resistant to these drugs? What new therapies do we need to develop? Do we approach these diseases As chronic diseases, you can give ibrutinib for years, and patients are in a partial remission for years, and they feel just kind of fine, but they're taking a drug forever. Or is it better to define strategies that will cure them, and so you don't have to do that? I know what I would do. I'd rather cure the disease. And how do you reduce the duration of treatment? Right now, you got to give ibrutinib, idelalisib, ABT-199, IPI-1 forever. And they're reasonably well tolerated, and they're all pills, but they're expensive and they're forever. Since it's getting late, I'll conclude, sort of. There's a few more here. We are moving to a chemo-free world. To do this, we have novel agents that target the cell surface of the tumor, the monoclonal antibodies, the intracellular pathways, the kinases and pro-apoptotic drugs, the ibrutinibs and idelalisibs and the ABT-199s, and the microenvironment, the IMiDs such as lenalidomide and the PD-1, PD-L1 drugs. We have to recognize the heterogeneity of these lymphomas and develop rational combinations of drugs leading to individualized, personalized therapies. To get to this point, we have to accrue patients to clinical trials. This will increase the potential for cure. So I envision a world where instead of getting chlorambucil, instead of getting bendamustine, which even though I brought into the United States, I just as soon see disappear, being replaced by non-chemo. We'll have a monoclonal antibody, maybe have another one. I showed you some data with that before. We'll have drugs that target the BCR, the B-cell receptor, such as those that target BTK or PI3K. We'll have drugs that are called pro-apoptotic drugs that stop these cancer drugs from surviving as long. We'll have those that affect the microenvironment and put them together in a rational fashion. We may eventually be able to cure them, but it's going to take a change in how we think. We're going to have to put together the patients, the advocates, the pharma companies, insurance companies, because these are really expensive therapies, the medical and scientific community, and the government. But if we do this, I think we're going to finally see the end of chemotherapy. And I have to throw this in for your benefit. And to do this, we're going to have seeds of hope to plant the grass around it. But until we do this, this is the serious part now. Get serious. This is cancer. Get serious here. Very serious. In order to do this, we can actually prevent these diseases. We can prevent them. And I found a paper in my favorite journal which shows me how we can prevent all these awful diseases. In the Wine Spectator, my favorite journal, long-term wine drinking linked to low lymphoma death rates. So I submit to you tonight, when you're done with this dinner, go and treat your lymphoma. And live long. Thank you.